In the Transkingdom RNA interference or RNAi procedure, bacteria are used to deliver short hairpin RNAs or shRNAs targeted against the gene of interest, MDR1 in this protocol, into cancer cells with the aim of silencing the gene of interest by RNAi. First, Escherichia coli bacteria are transformed with an shRNA expressing vector. Beta-1 positive cancer cells are then seeded so that the confluency is 70% to 80% 24 hours after seeding. In the next step, the shRNA expressing bacteria and the cells are co-incubated for 2 hours. Finally, the co-incubation is terminated by antibiotic treatment of the cancer cells. The results show a downregulation of the gene of interest on the mRNA, protein and functional levels through real-time PCR, Western blot analysis, cell proliferation assays and drug accumulation assays.
The main advantage of this technique over other standard procedures like siRNA transfection using oligofactor is the effective and the direct delivery of siRNAs into the target cells.
Transkingdom RNAi or tkRNAi technology uses non-pathogenic Escherichia coli to produce and deliver therapeutic short hairpin RNA or shRNA into target cells to induce RNAi. A first-generation tkRNAi mediating vector or TRIP contains the bacteriophage T7 promoter for expression regulation of a therapeutic shRNA of interest. Furthermore, TRIP contains the INV locus from Yersinia pseudotuberculosis that encodes invacin, which permits natural non-invasive bacteria to enter beta-1 integrin-positive mammalian cells. TRIP also includes the HLA gene from Listeria monocytogenes, which produces listeriolysin O. This enzyme allows the therapeutic shRNA to escape from entry vesicles within the cytoplasm of the target cell. TRIP constructs are introduced into a competent non-pathogenic Escherichia coli strain, which encodes T7 RNA polymerase necessary for the T7 promoter-driven synthesis of shRNAs. The bacteria and eukaryotic cells are co-incubated, allowing the bacteria to enter the eukaryotic cells. The shRNAs are released and enter an RNAi pathway. To begin this procedure, grow a culture of E. coli bacteria transformed with the shRNA expressing TRIP vector with an overnight culture as described in the accompanying written protocol. On the day before incubating the bacteria with the target cells, seed 25 × 10 to the 4 human gastric carcinoma cells per well in 6 well dishes. The number of seeded cells depends on the speed of cell growth of the cell line. Plan the seeding time so that the confluency is about 70 to 80% 24 hours after seeding. Incubate the seeded cells at 37 degrees Celsius in a 5% carbon dioxide water vapor saturated atmosphere overnight. Before starting the cancer cell infection, measure the optical density of the overnight cultures of the tkRNAi vector-containing E. coli. Determine the OD600. Dilute the bacterial solution to an optical density of 0.5. "30 minutes before the bacterial co-incubation, replace the FCS-containing cell culture medium of the carcinoma cells with FCS-free medium. During the 30 minutes period, wash the bacteria twice with 1 × PBS by centrifugation at 4000 rpm for 5 minutes at 4 degrees Celsius. And resuspend in serum-free Leibovitz L15 medium to the original diluted volume. Once the bacteria are resuspended and the cells finish their 30 minutes incubation, add the bacteria to the cancer cells at the desired multiplicity of infection, or MOI, which should be in the range of 1 to 300 to 1 to 700. Co-incubate the bacteria and cancer cells for 2 hours at 37 degrees Celsius in a 5% carbon dioxide water vapor saturated atmosphere. After 2 hours of co-incubation, wash the cancer cells twice with 1 × PBS and once with serum containing Leibovitz L15 medium, supplemented with 100 units per milliliter penicillin, 100 micrograms per milliliter streptomycin, 2.5 micrograms per milliliter amphotericin, 150 micrograms per milliliter gentamicin, and 100 micrograms per milliliter kanamycin. Continue cultivating the cancer cells at 37 degrees Celsius in a 5% carbon dioxide water vapor saturated atmosphere. The effects of the bacterial treatment can be visualized by fluorescent microscopy, measured by quantitative real-time RT-PCR on the mRNA level, and examined by Western blot analysis. Functional assays like FACS analysis and cell proliferation assays can also be used. This representative image was taken 3 hours after incubating human gastric carcinoma cells with bacteria expressing therapeutic shRNA. Bacteria can be detected around the nucleus of the treated cell. In this experiment, downregulation of the expression of the multi-drug resistant gene MDR1 was examined by real-time PCR. Downregulation of MDR1 mRNA of about 70% can be seen after treatment with a therapeutic bacteria. MDR1 downregulation also took place on the protein level after bacterial treatment. MDR1 protein levels decreased over time compared with the control cell line. Functional analysis based on the results of the cytotoxicity assay shown here can be used as an indicator for the functioning of the Transkingdom RNAi. While a drug-resistant cell line does not respond to the cancer drug daunorubicin, the same cell line infected with bacteria transformed with an anti-MDR1 shRNA-expressing plasmid is sensitive to the drug. Finally, in an anthracycline accumulation assay, the cancer-resistant cell line showed little cancer drug accumulation, which was observed in the cancer-resistant cells treated with the MDR1 shRNA.
Once mastered, this technique can be applied within 28 hours if it is performed properly. Following this procedure, other techniques like RT-PCR can be applied in order to answer several questions like the regulatory effects on the gene of interest. After watching this video, you should have a good idea about how to apply Transkingdom RNA interference by co-incubating shRNA-expressing bacteria and beta-1 positive mammalian cells.